In the development lab, we're going to look at two parts of histology. The first part, we're going to look at the secondary follicle and all the different parts of the secondary follicle. However, I want to go over all the follicles with you. So we have an ovary here, and we have lots of follicles. So we have some follicles here, we have some follicles here. Whoa, that one's ovulating. So out here in the cortex, we are going to have follicles. The primordial follicles are the ones that are asleep. They're just going to be this little line, circle. They're going to have the primary oocyte, and again, oovocytes, the real pronunciation. And they're going to have simple squamosal epithelial around them. Now, we have the primary oocyte. These have been woken up by follicle stimulating hormone, and they're going to be the cell, and there is our primary oocyte in there, and the follicle is going to be cuboidal, putting red cuboidal cells around here. Now, I don't like this one. You can't really see the antrum. This one's ovulating. Our secondary follicle is going to be huge. We're still going to have our little buddy in here. There is our oocyte. I really love saying oocyte. It's going to be surrounded by some supporting cells, and we're going to look at those structures. There's going to be a fluid-filled antrum around it. Then there's going to be more supporting cells, and these are cuboidal, they're not circular, around it. And that's what we looked at last time in class. We have our primordial follicles, and they're out towards the outside of the ovary. And they're going to have just simple squamosal cells. And the oocyte is making up most of the space. We have our primary follicle here, and you have cuboidal cells. Now, these are woken up. They're going through meiosis 1, and they're racing. So this guy's actually racing with his neighbor right here. And only, hopefully, one will win. You have fraternal twins if two of them win. So we're going to look at the secondary follicle and actually associated structures now. So this is the secondary follicle. There's the oocyte. There is the corona radiata. There is the zona pellucida. There's the antrum. And that's how you tell it's a secondary oocyte or follicle. And then you have the outer cell. So here's a very nice secondary follicle, and we're going to draw this one. I tried to find some online for you to draw. I couldn't, as of making this recording, I couldn't find a really nice one. But here is the secondary follicle. And notice that's the primordial one way out there. This thing is humongous. So we're going to start with drawing the zona pellucida. And this actually has receptors on it that's going to help the sperm bind with the membrane and draw it in. Now, there's going to be cytoplasm in here and a nucleus where we could see the chromosomes, because the chromosomes are undergoing meiosis. So here it is. Isn't it beautiful? This is our primary oocyte, and these cells are undergoing meiosis. And it actually stops being a secondary follicle when it finishes meiosis 1. It becomes a tertiary follicle and starts producing lots of estrogen to be ready to be ovulated. Now, out around it here is the corona radiata. This is what the acrosome of the sperm have to actually work through, and it is stratified columnar epithelial. So I will now spend the next 5 minutes drawing stratified columnar. And 20 minutes later, I have my stratified cuboidal epithelial. And you know I cheated. I colored in first, and then I did the etching marks, right? So we then have a fluid-filled antrum. And I'm going to actually be lazy and let this be the fluid-filled antrum. And the presence of this antrum really separates this from the primary follicle. The primary follicle is literally the oocyte, the nucleus going through meiosis, and it's either going to have one or 2 layers of cuboidal epithelial, depending on how long it is around. So we're going to have the antrum. Then we have other cell layers around it. We're going to have 2 zones. We're going to have the zona granulosa and the theca interna. So the zona granulosa, that's the outer border of this antrum, and it contributes to it. The theca interna is going to be making estrogen in the follicular stage, and it will actually contribute to part of the corpus luteum after ovulation and make progesterone too after ovulation. So the theca interna is basically an endocrine organ. It's making estrogen that's helping with the increasing the lining of the uterus. And the zona granulosa is the outer zone. So we're going to put those both on here. So this inner lighter colored reddish is the zona granulosa. And I was laughing the other day reading your histology. Some smart student was just drawing lines like this to my own lines. I'm like, that is genius. So there's my zona granulosa. And then here is my theca. And that was allegedly a red color. It didn't really come out red. And there's my theca. So this is a secondary follicle. So this is our first histology. Now, when you're done with this drawing, I want you to go to my website. There is a page where I have both the histology of the follicles and enlargements from that model I just showed you. And you have to match the follicular stage with it. And let's just talk about the last 2 stages too. So we have 2 more. We have the corpus luteum, which is after ovulation. It maintains the endometrial lining. And then we have the remains of the corpus luteum as a corpus albicans, which is collagen fibers. What a surprise. And this goes away in a couple of months. Thank you.